Hello, I am Kishendran, and today our group is conducting a research reporting critique on a study titled Physical Activity is Associated with Gut Microbiome Features and Organic Acid Patterns in Adults Consuming Plant-Rich Diets. Our goal today isn't just to summarize facts, but to apply the scientific inquiry process to distinguish between weak and strong evidence. We want to understand how lifestyle factors like exercise truly interact with our internal biology, especially for those of us on specific plant-based diets. Moving forward, what is the importance of work in this matter? In the larger scientific community, we know that plant-rich diets are generally healthy, but they often come with unique stresses like chronic low-dose pesticide exposure and gastrointestinal sensitivities. This study enters the conversation by looking at the gut exercise axis. It explores how physical activity might just be the key to balancing these microbial systems, ecosystems. The author specifically hypothesized that meeting the WHO threshold of 150 minutes of activity per week would correlate with a significantly more robust gut profile. As your lead presenter, I want to drive home the big picture. When we look at this study, we are evaluating the quality and accuracy of reporting. Is this study just a collection of data, or does it provide a logical argument for a global society? The authors aim to provide a roadmap for personalized nutrition and fitness. However, as we move through our critique, we will ask the most important question in scientific inquiry. How do you know? My teammates will now dive into the specific evidence to see if these claims hold up to rigorous critique.
Thank you, Kishendran. To evaluate this work, we first look at the methods. The researchers used a cross-sectional design with 93 Romaine adults. These participants were split into two groups, those exerting more than 150 minutes a week and those exerting less. They used the IPAQSF questionnaire for activity levels and advanced 16S RNA gene sequence to identify bacteria in those samples. For metabolic tracking, they utilized the HPLC to analyze organic acids pattern in urine. This multi-omic approach shows that the researchers were aiming for a comprehensive view of the body response. So, what did they find? The higher PA group showed significantly higher microbial richness, specifically in beneficial taxa of Faecalibacterium. They also found distinct patterns in metabolites related to mitochondrial function. The authors concluded that the physical activity is the primary driver of microbial diversity. However, while the reporting is accurate, the strength of this evidence is where our critique begins.
Our first subsequent focus is the research design. The study is cross-sectional, a more snapshot in time. In the scientific inquiry process, we must distinguish between the association and the causality. We cannot prove that exercise caused this microbial shift. It is just as likely that people with a healthier microbiome will have more energy to exercise. Without a longitudinal study, the advice remains weak in terms of proving a direct cause and effect relationship. Secondly, we must for the measurement method, the actor relied on self-report questionnaires, IPAQ, SF. This introduced record bias. People often over-report their exercise to look good or simply forget the detail. As we discussed in class, occurrence in reporting is antisocial, using objective tools like smartwatches would have provided much stronger advice for scientific community.
Building on that, we noticed a significant deficiency in the sample. The lower PA group was significantly older than the higher PA group. Since we know the microbiome naturally changes with age, the authors may be misattributing age-related changes to exercise. This is a questionable generalization. If the groups aren't age-matched, the accuracy of the conclusion is compromised. Looking at the global society, this research suggests that physical activity could help mitigate the negative effects of modern dietary stressors. However, for subsequent scientific exploration, we suggest that future researchers move toward randomized controlled trials and include more diverse global populations beyond just this small Romanian cohort. To sum up our critique, while the study is well prepared and uses high-quality lab tools, its conclusions are limited by its design and subjective measurements. It is a valuable starting point, but not a definitive answer. As a team, we've learned that in scientific inquiry, the tools you use are only as good as the logic behind the study design. Thank you for your time.